lecture so that your friends um, can have access to the recording if they are not able. Okay, by the way, what do you see in the screen at the moment? Is it the ELIP with the um, file explorer, or is it the advanced genetic page, PowerPoint slide that you can see now?
The advanced genetic.
Okay, so the PowerPoint, right?
Yeah, yeah.
Okay, right, because I am using two screens, so I think I'm sharing the screen number 2, which is the advanced genetic, but somehow the other part of the WebEx appear on screen number 1, so that's why I'm not that sure. Okay, this is course STB2122 Advanced Genetic for academic session 2021-2022. All right. This course is actually a continuation of your STB 1033. So it is more or less what you have learned from STB 1033 will be carried on further in this course. And then sometimes during the course, I might want, or I sometimes will ask question, probably simple question related to genetics, which most of the question that I ask are from what you have learned in STB 1033. Okay, so that just a question to see or to gauge your understanding on certain topics. All right, but it is not like I'm evaluating in terms of using it for marks for your for this course. All right, but once in a while, probably after 1 or 2 lectures, I will do a quiz. Okay, so, and that quiz will be accounted for towards your final grade, right? You remember later I will explain further in terms of the percentage distribution throughout the semester for the evaluation purposes, right? So some of you might have met me before in 1033. Some of you meet me first time in this course. So my name is Associate Prof. Dr. Muhammad Hasnan Hussein. You can call me Dr. Hasnan. If uh, you can always contact me by email over here. Um, and also, if you are within campus, most likely you are not now, except for some of you who has come over to UNIMAS for certain circumstances, which you are allowed to be in campus, but you are still online. But so if you are in UNIMAS, you just dial within university. If you use the university-wide phone, it is 3012. Okay, if you are calling from outside, 5830. 12. I am located in room G2 of Faculty of Resource Science and Technology. You can always come and see me later when most likely in the second semester you'll be attending the course in person. Okay, but at the moment, everything is through online. I will create a WhatsApp group for everyone. I haven't decided yet whether I want to create separate group for G01, G1, G2, and G3, or I just want to group every one of you together in one large WhatsApp group. Okay, that one I haven't decided yet. All right. But feel free to contact me. At the moment, you can always email me, or you can always uh, contact through the ELIP messaging system. But with the ELIP messaging system, sometimes I might miss the the messaging there, because it is only when I open the eLib, I can see the messaging system there. Okay, but you can always email me. All right. As I mentioned before, coverage of genetic concept, further extension of what you learn in general genetics. So there are several areas in genetic that you will learn further in this course. Gene function, genomics, and then you will come across quantitative genetics, in which you do a little bit of calculation that you can relate to genetics, right? And then gene mapping, in terms of once you do genomics, then you before genomics or you can see that some other researchers or scientists before this long time ago, they do mapping in terms of determining certain characteristic, and then relate it to location on the chromosome. Okay, and then how genes control gene regulation, and also how the evolution is also influenced by genetics. Okay, if you learn about the Darwinian theory, survival of the fittest, and so on, adaptation of organisms. So those are also can be explained through genetics, how changes can occur. So this one is under molecular evolution, right? So this aim of the course, what I can say is that at the end of this course, you can always revisit the aim of this course, whether when you read all these aims that we list over here, you are able to visualize something or not. Okay, but it's not necessarily that you know in a very great detail in terms of what are all gene functions. Okay, and then you did not know in very detailed quantitative genetics, gene mapping, and so on. But when you read through this list, you can visualize something related to what you have gone through in the course. Okay, so this aim of the course, and towards the end, you can try and revisit this and see whether what we aim to provide you throughout the semester, you gain anything or something, or a lot based on this course. Okay, so there are 4 LUs: gene function, genomics, quantitative genetics, gene mapping, and regulation. All right, so pronunciation also becoming haywire because we haven't been giving lecture for quite some time. All right, so 4 LUs, and all the contents in the aim are covered in this LU. Okay, now come to the assessment. The assessment will be the final grade, 100%, right? So what constitute of this 100%? They are 30% assignment and quizzes, mid-semester exam. There will be one mid-semester exam and one final exam to the end of the semester. Okay, and assignment and quizzes, we'll see the scheduling. We try to follow the scheduling based on that. The quizzes, I have designed, or I have already set several quizzes. So throughout the semester, we have 3 or 4 quizzes, and then a few assignments for you all. And we might also need to do a practical, but this practical is a home-based practical. And those will come or fall under the assignment. So all those assignments, including the practical reports and quizzes, 30%. So what I normally do is that assignment will become 25%, and several quizzes, they will be 5% average of all the quizzes that we will do throughout the semester. Okay, and method of teaching is via WebEx and also ELIP if you are unable to attend the online lecture. With regard to the online lecture, usually the attendance will be taken. Okay, but I understand that some of you most probably wouldn't be able to be present throughout the entire lecture, and you have to out and then come back in. Don't worry about that because I will have recording, but try as much as possible to be online during the assigned lecture. Okay, in terms of, okay, before that, I have three groups for this course, okay, G1, G2, and G3. What I do for the online course is that instead of I am repeating, for example, actually for G1, the lecture will be supposed to be from 8 to 10, okay, and then 10 to 12 will be G2, and then on Thursday 2 to 4, there will be G3. So instead of rather than repeating the same lecture live for every lecture group, what I have in mind is that I will be doing one live lecture like we are having now. For today, I started at 9 o'clock until 11, in which I cover G1 and G2 timing. Okay, some of you might be able to leave after 10, so don't worry about that. You can leave and continue with the lecture after you have free time. You can watch the video. So in terms of attendance, most likely you are unable to fulfill. Okay, for example, the G3 might be unable to attend this lecture. So you can always watch the video, right? And another thing is that I haven't decided whether I want to do all live lecture at 9 until 11 every week, or we'll do, for example, the first 4 weeks is between 8 to 10, and then week 5 until week 8, I will do 10 to 12, and then week 8 to week 12, I'll do on Thursday 2 to 4. I haven't decided yet, but what I plan to do is that I will um, um, do another quiz like you did before, based on the quiz that you have, no, not the quiz, sorry, the survey that you have answered, the questionnaire that you have answered. It looks like this time around, quite a high percentage, about 30, more than 30% unable to join the lecture that I am having live at on Tuesday, for example, that I try to set. Okay, but because in the previous year, more than 90% are able to attend one of the timing that I have set, even though their lecture is at different time, right? So for this semester, it looks like for your lecture groups, if I do at certain time, quite a big percentage unable to join for that time. All right, so I will do another questionnaire afterwards, and then you you please answer to that. I can gauge how can I proceed for the second week onwards. Okay, all right. In terms of reference, this one, unfortunately, you are out of campus. You are unable to access the books in the library, but these are the main books, or this is the main book that I use for the lecture content. Okay, but there are other references book. This one is a very good Jin. Now it is not Jin's 8, it's a Jin's, I can't remember, 10 or 11 already. So, but if you type Jins and then Benjamin Lewin is the author, then most likely they will have some notes online. So this one is a very good book. You can even use this until your postgraduate if you later want to do masters or PhD. This one is a very good book. Alberts and Brooker are also good books that you can do references. Okay, so you can search online for some of the lecture notes or some of the uh, extract from the books that we also use in the lecture. Okay, so that will be the what I plan to cover for our lecture, right? Okay, the next one will be our lecture schedule. I'm going to share that as well. Okay, before that, any question? Okay, in the chat, some of you mentioned that you have to leave the meeting halfway because have class at 10 a.m. Okay, don't worry. As I mentioned before, if you are not actually in the actual slot for my lecture, and then you have to leave for your other lecture during the time that I'm taking for this lecture, go ahead. Uh, please leave. There is no marks deduction or any penalty whatsoever. I understand that. You can always come back later when we finish this lecture. I will put this online. Okay, the recording I will put in the e-link. The link for the YouTube I will put in the e-link. So don't worry about that. For the attendance, okay, I will key in afterwards. May I ask for this course, why is the credit hour is 2 and how does it differ from other core courses with 3 credit hour? Okay, 2 credit hour means that every week you are supposed to have only maximum 2 hours lecture. Okay, so 2 hours lecture is usually actually there are, if later on I will show you the course plan afterwards, yeah, after this, after the scheduling. For your general understanding, for courses, whatever courses that is 2 credit, okay, usually, okay, but not it is not a rule, okay, but it is a guide. Usually for 2 credit courses, it is 2 hour lecture maximum. And 3 credit courses, you will have 3 hours lecture in a week. Okay, so that's why if you are also taking other core courses, you will see that for your 3 credit courses, for example, 2163, for example, you will take in the second semester, I'll be teaching that. 2163, you will have 3 lecture course, but then sometimes that 3 lecture course, if it is a practical classes, it involves practicals uh, um, many times. So you can have 2 hours plus the practicals, several hours of practical will constitute of 3 credit hours. Okay, so that will be the reason why uh, the question why credit hour is 2 and how does it differ. So the difference is in terms of lecture, and later on when I will explain in the course plan, you will also find in terms of the learning hours. Okay, so for 1 credit, it constitute of 1 lecture, uh, 1 lecture hour, and then 2 lecture that you have to spend on your own doing a revision related to that particular lecture. And then you will have another 37 hours that will actually cover the other activities. For example, you have quizzes, So in order to do have the quiz, you need time to study. You need to spend certain time to study. So that will count into that as well. And then a mid-semester exam, you will have let's say 2 hours of mid-semester exam. So you need another 4 hours of study time on your own free time. So basically, every 1 credit, there are a total of I forgot what's the term, 40 hours notional, 40 notional hours in which you are supposed to spend for your for this course. Okay, so that is why uh, 1 credit, meaning to say 1 hour lecture plus 39 hours of something else throughout the semester. So 40 hours per credit. Okay, it might confuse you a bit, but for your knowledge, you can always assume that 1 credit, 1 hour lecture. So this course 2 credit, so 2 hour lecture every week for 14 weeks. Okay. All right. The attendance I will key in for those who are unable to scan. Actually, I will show again the QR code afterward. Where were we? Okay, the scheduling, right? So for the scheduling, I will share the scheduling now. Where or what? Okay, Microsoft Word. Okay, so this is the lecture schedule for semester 1, 2021-2022. Okay, similar like in the previous year. So what I usually do is that I will have lecture 1 until lecture 12. So sometimes because of mid-semester exam, I will use the lecture time. Then that lecture will be moved to the following week. So we'll do until week 13, for example. So we'll have 12 lectures, hopefully in 13 weeks. And then if somehow during this time, I have to, um, let's say, unable to do the online lecture, then or sometimes it got dragged too long, then we'll cover until week 14. Okay, but I will always try to finish everything by week 13. Okay, and then for you this semester, I noticed from the academic calendar there is no revision week. So that is also why somehow we can use the week 14 as your revision week, because for this semester you will have 14 weeks of lecture followed straight away by examination weeks, 2 weeks if I'm not mistaken. Okay, so this one is also covered or will be given to you. So you will have assignment 1. Okay, and then you will have work from home practicals to be confirmed. What are the activities that you will be doing, and then you have assignment 2 and assignment 3. So more or less in total there will be 4 activities constituting of 3 assignments and 1 practical. So they will be average everything 100% for that specific activity, and then converted to 25% average for all. Will then contribute to the assignment and quizzes, okay, of the 30% over here. So 25% of assignment, which constitute 3 plus 1, and then quizzes, several quizzes. So that will be what is going to be covered in your course. Okay, what else? Okay, a very short cost plan, and I just want to share with you cost plan and also this one. What is this? This one is your scheduling. Okay. Okay. Hang on, yeah. Plan.
Excuse me, doctor, I have a question.
Yes. 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 Please.
May I ask if you would like to join the G02 WhatsApp group for advanced genetics?
So you have created a group for G02 only, correct?
Yes, sir.
Oh, okay. So in that case, actually, how many participants can be in one WhatsApp group? It is less than 100, isn't it?
Up to approximately 200 odd, 250 something.
Oh, okay. Um, I I think what I'll do, I can join that, but then I will also, I haven't decided yet, okay? But then what I thinking along the line is that you can have that group or the other group can also have G1 and G3, your own WhatsApp group in order for you to discuss something. But then I would prefer at the moment what I have in mind is that I have only one WhatsApp group for everyone. The reason being, if I type something, I want to convey something, everyone can have access to my message. You see, at only one time I do the messaging. Otherwise, I will have to forward the message to two other groups, right? Or some I might forget that. Um, at the moment, that is what I am inclined to do. One big groups in where everyone are in the groups. Okay, but then I can also be in the G2 group. Um, the other reason is because sometimes you might want to ask question related to the subject, and I will answer to that in the WhatsApp, and uh, I would prefer if every others can have access to the answer so that you can learn something from it. Okay, and if you ask question about assignment or whatever, I can type only once and then everyone get the message. Would that
I
be
see.
okay?
All right. Thank you, sir.
Okay, but it's okay if you want to invite me, but you have to also consider, because I know some of you don't want the lecturer to be in the group. You want to discuss about something that you don't want the lecturer to know about it, kan? Then you discuss among your friend lah. Okay, okay. You discuss
All
among
right.
yourself first what you want to do. But I understand that. I know sometimes because I am there, then you have to create another WhatsApp group just for you to gossip or whatever you don't want me to know. Kan? Okay,
Yeah.
boleh
Okay.
ya?
Thank you, sir.
Okay, no problem. Okay, all right. Advanced. Where were I just now? Oh, sharing the cost plan, right? Okay, okay. We'll do this rather quick. I don't know. Is this? Wait, yeah. I try to share again. One. Okay, now I'm sharing a screen one. You see the cost plan, isn't it? Can you see it? Hello, everyone. Boleh
Not
nampak?
yet, doctor.
Huh?
Not yet.
No,
Not
no,
yet.
No,
Oh,
doctor.
no,
not yet.
doctor.
Okay, can you see now?
Yeah, yeah.
Yeah.
Can see
Okay.
now.
All right, so this one is cost outline. I will put in the ellipse as well. So these are all the administrative things, your PLO, CLO, you don't necessarily need to know lah, but we have to show you as well. So usually for teaching and learning, okay, this one is the one that to follow up to what were asked earlier, okay? Student learning time, 67 hours. So actually, if you include everything, grand total of 75, and actually, and then you include all the exam, everything, it comes to 80 student learning time, including the exam time. So that is 80 hours for 2-hour credit, okay? Later on, you go to other courses, 3 credit, you will see that the total will be around on average it's 120. So these are the total hours that you were asking about the credit. So every one credit constitute of 40 notional learning hours. So everything will be 80 hours. Okay, so this one is the course outline, mid-semester 30, 40. You can see over here. So like for example, student learning time is 4 hours. Non-face-to-face 2 hours that you have to learn, and then physical 2 hours. So this one is actually the one that I haven't updated. Only now I realize it, because you will be non-face-to-face here. This one, these 2 hours actually should be over here. Okay, online technology mediated for your final exam, because you are not in campus, isn't it? To do the exam. So this one will be face-to-face, synchronous online live activities. So this will be 2 hours over here, and this also. So there is no physical activity. Okay, so these are the what do you call that? The course outline. And then another one is course plan, similar activities. So also you can see over here explanation of CLO, course learning outcome, everything, the one that I explained to you, the aims, everything. So it falls under learning outcome, course learning outcome. And then you have learning units, so activities, lecture, discussion, like we are having now. And then cybergogy is something related to what you learn through online access to whatever information that you can access online. Okay, cybergogy. And then pedagogy, something like you learn actually on the topics through peer groups. Okay, heutagogy, I actually don't know about it. Okay, I read about it, but I forgot. Okay, I can only remember these two at the moment. Okay, if you want to know further, you can ask me later, and then I can go and find it and then inform you, or you can do it yourself. Okay, but this one is the new terms and I'm not familiar with. But then what is inside there? Usually we are quite familiar with that. I'm quite familiar. For example, the one that we are using, I didn't realize that it was termed cybergogy and so on. Okay, so that will be it in terms of course plan. So all right, so now we will go into our lecture 1, gene control. We have introduction. I have completed that. Okay, gene control of proteins. Okay, okay, so I'm going to share this. Bear with me for a few seconds, yeah. You full screen mode. Okay. All right, and then I'm going to share into share. All right, gene control of proteins. Actually, as I mentioned earlier, iGenetics Molecular Approach second edition. So that is the main book that I use for this course. The reason being iGenetics way of describing some of the theory related to genetics are very very, how do you say that, not layman's term, but easy to understand. It's not too much theory until you got lost when you read it. Okay, the way it explained in the book is very actually it's not layman's, but because it is a textbook, but the way it explained is quite easy for us to understand. All right, so that is why I quite like this iGenetics by Russell, Peter J. Russell. So you can later on you can search online cyberogogy. Okay, you can search online and see the contents. But in the library, we have one. I recommended library to buy several copies of the book. Gene control of the enzymes. This one is just a basic one in the cell. You know that this one is this one prokaryote or eukaryote? Prokaryote or eukaryote? Anyone? You can type in the chat. You know, prokaryote or eukaryote? This one, okay, I can see one or two. Okay, good. Some of you have answered eukaryotic cells, isn't it? Because there is a nucleus there. So in the nucleus, there are chromosomes, and then in the chromosomes actually constitutes of DNA and what's another one? DNA and something else that make it into a structure. So, and then within the DNA, there's a part where there are genes, okay? And then these genes are then what you call converted into mRNA, copied into mRNA, transcribed, and then RNA are processed and then exported outside the nucleus, which will then be translated into proteins, and the proteins can function all over the cells and transported somewhere else, right? So what are the components that make the chromosome is the protein, isn't it? So go back and do some revision. The proteins, the histone proteins and so on. So, but we are not talking about the structure. We are talking about what controls the gene, the enzymes. So these are the mechanism that you can visualize. So DNA has gene. Gene will produce, will transcribe into mRNA. mRNA will then be translated. So those translated molecules, translated macromolecules, will then be used for functions, okay, proteins, and then some of them can go back into the nucleus, transported back into the nucleus, in which it will control the process of producing the mRNA for other components. All right. For example, have you come across the purple tomato? American tomato. Have you come across purple tomato? Yes or no? No.
No, doctor.
No, ya. Purple tomato, salah, American pula. Purple tomato, Melayu punya pronunciation, actually is one of the example of a plant where the gene control is manipulated to produce the purple color. I'm going to show you whether I can find it or not. So what actually being controlled is that it is not the gene for the color. Okay. All right. I will show you. I'm going to show you. To stop sharing for a short while. Okay. I'm going to share with you. Okay. This one. Okay. All right. So you can see a lady over here, right?
Yes,
Yes,
doctor.
doctor.
All right. So this lady actually, my former supervisor when I did my PhD about 20 years ago. Professor Kathy Martin. I did my PhD at this place, okay, John Center in Norwich, UK. So, what you can see on the table in front of her are fruits and vegetables, okay, that constitute a component that has, for example, additional nutritional effect. And this one, for example, the cauliflower, and then there are like tomatoes. So what happened is that what they found out even when I was working with her a long time ago. Some of the other group, I work with starch synthesis gene, but her other group, my lab mates, they were working on transcription factor. So this transcription factor is not a gene that produce discoloring, okay? But this transcription factor is the one that control the production of other components that will produce coloring, the pigments for these fruits and vegetables. Okay, so what they did was that they didn't manipulate the gene that produce the pigments that give this color, which has a high antioxidant content and so on. So what they did was that they found out from the research that there is another component they call transcription factor, in which this transcription factor, they will come to the DNA when it is being transcribed, and it will allow for the mRNA for the gene to be expressed in higher amount, be transcribed in a higher quantity. So when you have higher quantity, then more pigment will be produced. So the gene itself is not manipulated, but you manipulate the other gene that control the gene that produce the pigment. Okay, so that is what the example of gene control in the enzyme. So now you can imagine that in our chromosome, when we produce something, whether produce the pigment for our skin color, eye color, hair color, and so on, it is not only the gene that produce the pigment. There are actually other genes that produce another type of protein, but small protein that can come and be around the gene that produce the color. Okay, so that will be produce more mRNA of that color gene and so on. So this is one example of the work done in Kathy Martin's lab. I just found out that she won this prize, so that's why I know about it. But I know about her work a long time ago, of course, because she was my supervisor a long time ago. But then her work is not only related to the fundamental of science, but can also be edible. Okay. Are the fruits edible? Yes, the fruits are edible. So the only issue that, but in the Europe, I think it's okay. You have heard about genetically modified organism, isn't it? So food like this can also, if you manipulate the gene by having a vector, put it inside, and then that vector will go to the gene and change the gene, then you have a genetically modified organism. But if you have a certain way of changing the gene without having external influencer, kan macam kamu duk tengok Instagram banyak terpengaruh dengan influencer, right? So in the gene also, for the gene also you can have influencer like that. I mean, if you have external gene DNA like the plasmid that go into the cell and change the content of the chromosome, then it'll be GMO. But if you can manipulate, there are certain ways to manipulate without it having without using the external influencer, then you can have it. You don't call it GMO, okay? But to answer the question just now, is it edible? Yes, it is edible. You can you can eat that, okay? So that is you can see over here, from DNA transcribed into mRNA process to remove the pre-mRNA, to remove the introns, and then it is translated. So the translation, the product of translation, not necessarily be the end product. Okay, for example, like in this case, it is a transcription factor is the product. So this will transported back into the nucleus, then it will influence the other gene, then it will produce at a higher quantity. So that is why it is considered as gene control of the enzyme. Okay, so that is how it works. Okay, do you think that? Hang on. Let me. Okay, Freddy. Thank you. I will talk to you later. Yeah, I will let you. Okay. So a colleague of mine passed some rice grain for my research. Okay. Do you think in future supplements that can change the amount of pigment you want in the eyes or hair will exist? I would say, you know, it is not impossible. Okay, it will not impossible, but how long does it take? Uh, I don't know. Okay, but I believe uh, some sometimes in future, I don't know whether it's going to be near future or very a long way ahead. I don't know whether how long does it take, but I believe it will, because you just imagine uh, genes are able to be manipulated, as we all know, to produce a set higher amount or lower amount, or you can switch off. That one, even now, we can always do it in my lab. Some of the work that we are doing, we can switch off the gene, just like the COVID-19, the SARS-CoV-2 virus, right? The vaccine for the COVID-19 mRNA vaccine. So long time ago, even when I was doing my PhD and slightly before that, the mRNA technology for that is quite we say that is quite recent. But then, if you say that mRNA can be used as a vaccine, nobody will believe you. If let's say 20 years ago or 30 years ago, let's say. The reason being mRNA is very sensitive. Like in our lab, we can extract the mRNA, but when we want to extract the mRNA, whether mRNA from human, plant, or animal, or even a bacteria, if we wear glove and then sometimes, even when you are wearing the glove, you have itchy hands, at the where the glove is not covered, you try to scratch with the glove, you scratch and then you work with you continue working with the mRNA. Most of the time, the experiment will not be successful because mRNA is very sensitive, very transient. Our skin got certain type of enzyme that can easily degrade the mRNA. So mRNA very difficult to be preserved or to be in a stable form. So if 20 years ago someone asked, can the mRNA be used as a vaccine? People will laugh. Oh no, not us. If someone say mRNA can be used as a vaccine, other people will laugh at you if you say that, because it is quite impossible. But then now you see what happened. You can always you know that mRNA is now used as a vaccine because they can create certain mice cells like this, okay, in which mRNA is inside, and outside they protect it with a certain lipid bilayer molecule, so it can be used as a vaccine. Okay, so same, coming back to the question someone asked just now, can you use a certain kind of injection or compounds that you can change the pigment? You might be able to do that, but you have to remember that, for example, the hair pigment and the skin pigment mostly come from the same gene. So if you want to change the hair pigment, your skin pigment will also be affected. That one is in a simple explanation. Okay, so that is one to answer. is it easy to commercialize this kind of finding, or is it can go as far as being labeled as lab product? So this one can be commercialized even in the certain countries. We can like soya bean GMO soya bean is already commercialized. So something similar like this, you can eat as a product that has been genetically modified. All right, someone has left the group. Don't worry, it's okay with that. You can tell them can access the lecture later on. All right. Manual attendance, yes, I will do that. Okay. Right. Okay. So those who have to leave because there are other commitment at 10 o'clock. Don't worry. Can always continue. All right. Gene regulation, transcription, post-transcription, processing, translation. This one you have learned before, isn't it? 1 and 2, transcription, in which the DNA are transcribed, and then this one is a pre-mRNA removal of the introns, and then they are spliced together using what? What is the molecules that splice the pre-mRNA into mRNA, mature mRNA. Do you remember that? Okay. Helicase is one of the enzyme that can cut the DNA. Okay, but not the helicase. Helicase got other function, which is also cutting the nucleic acid, but the DNA. Never mind, you can keep answering. I will inform you later at the end of the lecture. But you have to remind me about that. Okay. So and then the mature mRNA will what you call that exported outside. You remember this one is a lipid bilayer, and then there are proteins that can function as a tunnel. Okay, for transport. So there are this protein recognize specific molecules. So this one allow for mRNA to go through. So all cells mechanism is something similar like this. So if there are some other protein which recognize that specific, they will allow depending on the type of protein. So so they will only allow specific molecule based on the structure of this uh, uh, protein, which is located in between the lipid bilayer. Okay, so mRNA splicing and then mRNA protection by this 5' cap and poly-A tail. So you also learn about the 5' cap poly-A tail. After the gene is transcribed, addition of the guanine in reverse position, and then the A poly-A is added. So this one will make the mRNA is stable, okay, and then break down by small interfering RNA. Sometimes there are additional RNA that the job is to destroy the RNA, for example. So that one is another way of gene control. So you remember gene control not only in terms of it producing what you call that proteins, but there are also gene control using the RNA, silencing RNA. If you can, if you come across that before, when you were reading something, silencing RNA. So that is one of the way to control a gene. Okay, spliceosome. Well done. Those who answer spliceosome that manipulate or remove the introns, spliceosome. Okay. So, and then translation and post-translational modification. So this one is also important, but we don't learn it very detailed in this course. So you have to visualize that once the protein are being translated using the mRNA information, most of the time, the protein in our system or in the eukaryotic system, they are further modified. It's not like modified by cutting the protein and removing it. That happened, but usually the removal is at the beginning of the protein, and after that it will add certain molecule like lipid, like sugar molecule, and so on. So that one is called post-translational modification, which allow for the protein to be a functioning protein. Okay, not all protein are like that, but some proteins have to be modified in order for it to function. Okay. No question yet. What is the function of the helicase? Helicase, something related to helix, right? So it will remove the helix structure, you know, of the DNA. DNA is a helix, so it will unwind the DNA. That one is helicase. Ligase, you all know, isn't it? Okay, genetic control of the enzyme refers to controlling transcription mRNA needed for enzyme synthesis. Involve induction or repression of the enzyme synthesis by regulatory protein that bind DNA and either block or enhance the function of the RNA polymerase. So this one of the way, as I mentioned about, you remember the purple tomato just now. So that one is the regulatory protein that can bind the DNA and enhance the function of RNA polymerase so that the enzyme that is being transcribed with the presence of this regulatory protein are increased in terms of the amount. Okay, so that one is gene control that enhance. Okay, but if you block, then you repress. If you enhance, then you induce to make it to produce more. Okay. Regulation of gene expression or gene regulation, sometimes it is called, include a process that cell use to regulate the way that information in genes turn into gene product. So you just imagine you have DNA. Somehow I wouldn't be able to. Let's see whether I can annotate here or not. Okay, you have DNA, and then on the DNA, you have certain molecules. No, I need to set my my pen later. Okay, so for this time, this lecture, I wouldn't be able to do that. Okay, the information into gene product. So you can have certain molecule that can influence the production of mRNA for certain other gene. Protein expression is a subcomponent of gene expression. If you visualize transcription, translation, so protein expression is also the translation process. Okay, so sometimes you can use the term interchangeably. Probably I mentioned translation, and some other times talking about the same thing, but I use the phrase protein expression, actually referring to more or less the same thing. All right. Protein expression consists of stages after DNA has been transcribed into mRNA and then translated into proteins. Okay, gene control. So this one, okay, encode proteins. Genes work in set to accomplish biochemical pathway. Genes often work in cooperation with other genes. Just like I mentioned, you have two genes, two genes, one at the end here, there's another gene over here. So this gene actually is used to influence the production transcription process of this gene. Okay, so work in cooperation. So these are the foundation of modern molecular genetics. So what happens if mutation happens then? It depends on the mutation that happen where. The one that control the other gene or the mutation of this gene as well. Okay, you can, you know that mutation can happen in many ways. Mutation on the DNA, the nucleotide itself, or mutation on the DNA itself, right? So many ways mutation can happen. So in human, example shown here is something related to inborn errors of metabolism. Alkaptonuria, human trait characterized by urine blackening upon exposure to air, and some of these later in their life can have arthritis when other people in their age group do not have. Okay, so this one is alkaptonuria. So Archibald Garrett and William Bateson, 1902. So almost how many 100 years ago, 120 years ago. Okay, Garrett and Bateson concluded that alkaptonuria is genetically determined because when they did the study, families with alkaptonuria had several affected members, and alkaptonuria much more common in first cousin marriage than marriages with the unrelated partners. Okay, first cousin marriage is that the great grandparents have siblings. Those siblings have their own family, and those family have their family as well, and those family got married. So first cousin, and usually it has more instances of alkaptonuria compared to those who are unrelated. All right. So you remember William Bateson? Uh, what does William Bateson contributed in terms of if we talk about genetic? Anyone remember STB1033? William Bateson and genetics? Not only his work related to genetic, they actually study Mendelian genetics, but in other organisms. Okay, for example, they study in birds, in chicken, and show that it is similar Mendelian pattern, for example, kan Bateson. So what is another quite significant contribution of William Bateson in the field of genetic? Ada siapa-siapa ingat William Bateson? Tak ingat? Cannot remember?
Heredity.
Heredity, yes, something to do with heredity. But another thing is that William Bateson is also the one who coined the term genetics in one of his letter to his colleague. Right, he comes with the word genetic to um, to show something related to heredity just now. All right. And then when I was doing my PhD at John Innes Center, one of the building, actually the building that I work in is called Bateson Building. So that is William Bateson. So William Bateson is one of the modern geneticists lah, in addition to siapa? Gregor Mendel, Garrett. Okay. Normal and alkaptonuria, blackening of urine after exposure, and then ochronosis, the blackening of the white eye. Okay, and then some area on the ear, and then deposition of the pigment. Blackening pigment in the cell, okay, in some area of the tissue. So Garrett showed that alkaptonuria result from, so they did a biochemical study, and result we show that alkaptonuria in the urine, the blackening is because of the homogentic acid present in the urine. So Garrett reasoned that normal people, the homogentic acid is metabolized. Okay, but those that show alkaptonuria symptom, this homogentic acid cannot be converted to other molecule, and they remain in the urine, which then when you expose those urine containing homogentic acid, it will blacken the urine. Okay, so which means that the people that has the blackening of urine in their system, their body cannot convert the homogentic acid, and it is expelled out through the expelled out in the urine. Don't worry, it's okay. Those who have to leave, no need to type anything. It's okay, it's fine. Okay, for in future, just leave. I understand that. Okay. The responsible mutation is a recessive. The gene was later shown to be in chromosome 3. Garrett and Batson didn't work until the DNA and gene itself. Because remember, in 1902, do people know about the DNA already? Yes or no? Do scientists know about DNA in 1902? Anyone?
Yes.
Yes or no, chat? Yes or no? The answer is no, isn't it? The answer is no, because the DNA actually, yes, the answer is no. Yeah, because the DNA was explained by siapa? Watson and Crick, right? In 19 and towards 1950s, 1960s. So only then they know. So in between 1902 when they were working with this, and then towards identification of DNA molecule, how they looks like, yang all those experiment, what experiment that you learned before? Uh, Somehow I cannot recall this morning. No, the transformation experiment, right? Michelson and Stoll and so on. Okay, so those are in between when they suspect the molecule, hereditary molecule is the DNA. So the responsible mutation is recessive. So this one is kind of the timeline, chronological event when people know and the study until they know which part of the DNA. Blackening of urine, they discovered that it is a Mendelian recessive. Mendelian recessive means what? If you have 2 copies of the gene, we are diploid organism, isn't it? We have 2 copies of each gene. Then each gene can be either homozygous or heterozygous, right? So let's say it's a homozygous defective gene. Okay, so that metabolize homogenetic acid is defective, so it cannot metabolize both genes in the cell cannot metabolize, so it becomes homozygous recessive gene. So but what happens if the gene is one is recessive, the other one is dominant, which can convert the homogenetic acid, meaning to say that that individual will be a normal or abnormal alkaptonuric person. It will be a normal person, okay, because one of the gene is functioning, so this gene will be transcribed, although this gene is defective, okay. But if both are defective, then you call it a disease recessive gene. So please remember, again, just a revision, gene can be dominant and recessive, but if a disease is a recessive disease, it means that that gene that caused the disease exists in a homozygous form. Okay, so the recessive mutation is recessive that caused the alkaptonuria, then it means that both genes on chromosome 3, we have one pair of chromosome 3, both gene on both chromosomes are defective. Okay, so they and then later on, they map the alkaptonuric gene on chromosome 3 on the long arm of chromosome 3 close to the centromere, for example. Okay, and then they found that in Aspergillus, homogenetic acid can also be can have that similar gene, and then using the Aspergillus homogenetic acid, they are able to locate the, you remember this one is still a very big position. They do not know the DNA yet, but then they know on which chromosome. Okay, And then further scientists later on, after that, several years after that, on this region, they find where is actually the homogenetic gene using using the similarity between aspergillus homogenetic acid with the human genetic acid. Okay, so they find on which tissue you can find a lot of mRNA. You remember, throughout our body we have the same DNA, right? But then on our hand, the homogenetic acid is not being transcribed because homogenetic acid is not used by cells in our hand. So they take tissues from our body, from the sample. Okay, and then based on the northern blot, they find out that, okay, mRNA is actually the one that it imply that the gene is being used, because mRNA, when you want to use the gene, protein that comes from the gene, it means that the gene has to be transcribed into mRNA, and then that mRNA will be translated, isn't it? So to find if the gene is active somewhere in our body, we can find the mRNA, which then imply that the gene is being transcribed and also translated. So on different tissue, they take from the heart tissue, from the lung tissue, from your muscle tissue, whatever tissue that you can think of, they find that the tissue that contain the mRNA for homogenetic acid is the liver. So meaning to say that the liver is actually the organ that is is producing the enzyme that convert homogenetic acid to mallyl acetoacetic acid. Okay, so these are the study. Okay, and then this one they know the gene on the chromosome, but in detail, where on the chromosome then, what actually on this gene, the details of it. So when you locate the DNA, the position of certain differences that contribute. You remember at this time, during this time, belum lagi Watson and Crick describe the DNA, right? But they know the chromosome, okay? And they know that if there are changes at this position, it causes the alkaptonuria. So that means that scientists map the alkaptonuria related disease to this position. So this is what you call mapping. So next time, when someone or if I mention mapping of the gene, you imagine that this gene cause certain activity of producing something, and we map them to certain position on the chromosome. So that positioning the gene on the chromosome, that is what is meant by mapping, gene mapping. Okay, so you visualize that way. Okay, so gene that produce the color pigment, the melanin. Okay, now of course we know already because people already do the genome sequencing, but you just imagine we are in a world, in different world where genome sequencing hasn't been done. So we don't know where the gene is on the chromosome. So, but somehow there is a person who is what do you call that, suffering from albinism. So that albinism, when we compare the chromosome, these are differences on certain chromosomes. So it means that you map the gene that caused the pigment for our hair or skin on that chromosome. So that one is the mapping. So mapping is positioning gene for certain characteristic on the chromosome. Okay, and then once we know that, we can study further on that particular region. Then we study until, okay, we know that these are the DNA, the gene. So based on that, we can study further what is the sequence, and those sequence we determine how many introns and exons and so on. Okay, so those are for the study that has been done. and then now you can do PCR and so on. Okay, any question so far? In the chat, there is no question. Okay, chromosome location, as mentioned before, you learned before, 3q13.33, meaning to say on the long arm, the Q arm of the chromosome. You still remember why the short arm is called the P arm and the long arm is called the Q arm? Remember or not? A trivia, some of you in my class. Ingat tak? Why is it called Q arm? French word, yes. Okay, some of you remember French word, P arm and Q arm. So position 13.33. So this one, of course, they can position that. Okay, and then once human genome sequence has been done, okay, they take chromosome 3, they do the sequencing from the beginning until the end. So the molecular location is at this position, 120 million the nucleotide position until 120 20 million 500 something thousand. So you take this value, you deduct with this value, you can see what is the length of the uh, the gene, including the intron and exon that cause alkaptonuria. Okay, so this one the explanation for this. Okay, so I'm going to skip this. You can read, but basically the timeline of how those people discovered. So Garrett's work, first evidence of specific relationship between genes and enzymes. So remember the Garrett and Bateson, and then at the same time Masterson and Stoll, and then several others are also working to determine the molecule, which one is the hereditary molecule. So this one is more or less things that is uh, uh, synchronous or in tandem. People doing this, people doing that, related to genetics at the same time. Okay, but similar concept between them. Okay, so with the insight that mutation can block human metabolic pathway by damaging an enzyme, causing a detectable build-up of that enzyme substrate, he found similar relationship in 3 others human disease. So further work notice that changes on the position of the chromosome. We now know the gene over there cause certain diseases. Okay, and that is why it comes to the work done by Biddle and Tatum. So this one is the example. You can see the timeline. 1902 is in which Garrett and Bateson hypothesizing that this person suffers certain disease because cannot be he or she cannot convert certain chemical compound into some other thing because they don't have those capability due to their marriage, for example. So it's a hereditary. Okay, and then people like Biddle and Tatum, and later on some other people, they want to see the relationship between the nucleic acid and the metabolism, for example. So they use Neurospora crassa, which then eventually from their work, they determine that if you have this stretch of DNA, if you change that, it causes certain activity to be removed from the organism. Okay, and then they, based on this, they know that one gene, one enzyme hypothesis came out at that time. Okay, um, so this one is what is Neurospora crassa is all about. So in Neurospora, it can exist as a physical individual, whether it can exist in a diploidic form or in a haploidic form. Okay, for example, in human or mammals, for example, we as an individual only exist in the diploidic form, right? So in our cell, always have two copies, but we don't exist as individual in the haploidic form. We exist as a haploidic form, not as individual, but only in the form of sperm and ovum. Okay, so that is human. But in Neurospora, an individual of Neurospora can exist and thrive, live either as a diploid individual or as a haploid individual. Okay, so that's basically the thing about the life cycle that that can be used. So what happened is that when it is in the haploidic form individual, they will produce spores, and the spores can be grow to produce individual in the form of a haploid, right? So they can take a haploid. Why they want to work with haploid? Because with the haploid, you change one gene, it show as an individual that change already. Okay, so you do not need to worry about the other in the diploid organism. If you change one, the other partner, the other half of the pair can help to compensate, isn't it? But in a diploid form, the gene exists and in one copy. If you change that gene, meaning to say that there is no other gene, there is no pair uh, to compensate. So it exists as a individual. Okay, so this one is the diploidic form. So in the haploidic form, they can exist as individuals. So if you change one, you change the individual, right? So this one is the explanation. Uh, and another thing is that if they exist in one form like this, the big A, the big A will not mate with big A, it will mate with the small a and so on. Okay, so only crossing of big A and small a will produce a gamete fusion, producing a diploid individual, which will then undergo normal uh, uh, normal mitosis meiosis to produce 4 haploid nuclei. Okay, so based on this, they take the individual spores of the haploidic form to do the experiment. Okay, and then this neurospora can grow in simple minimal media, wild type. Simple minimal media means that like in human, if I give you rice and salt and leaf as a vegetable, we can live already. You can always modify rice using a little bit of salt, and then you tumbuk the leaf, become different food, so you can eat. So meaning to say you can generate different type of food for you to survive. Uh, for you to survive. Okay, but in some organism, in some individual, dia kena masak siap-siap, become nasi lemak. So only with the nasi lemak, the person will survive. Without nasi lemak, the person cannot survive. Okay, so that one, so the uh, the one who survive on nasi lemak, meaning to say that they need a full medium to survive. Okay, but so in a wild type neurospora, you provide a simple basic medium, whatever amino acid that is not present in the minimal medium, it can be generated by the neurospora itself to produce those amino acid in order for them to survive. Okay, but then in some other individual, in some other neurospora that is mutant, they cannot survive on simple minimal media because their individual cell cannot produce that amino acid. So we need to produce amino acid in order to allow for that mutant to survive. Okay, so that mutant is called auxotrophic mutant, unable to make a needed nutrient, and this mutant will only grow if the nutrient is provided in the medium. Okay, so based on this, they do a lot of mutational work. So they take this what do you call that? The spore, the haploidic spore. They generate a lot of mutants uh, by mutating the uh, the the spore with the X-ray, and then they cross with the wild type. You know, remember, the big A can only be crossed with the small a, so they cross with the wild type strain. So actually, they can, after after you have the seeds, you expose to the X-ray, you generate, you can see the changes. But that changes can be a temporary changes which is not hereditary. Okay, but what they want is the one that is the changes in the DNA that can be carried to the next generation. That is why they cross with the wild type, so that the whatever changes is retained in the DNA and then carried to the next. Meaning to say that the changes is in a stable form. So the cross ensured that effects were due to inheritance rather than direct damage to the radiation. You remember, when we are exposed to the sunlight, like in some of you, some of you I mentioned, you go to the beach, you expose to the sunlight. If you got moles, and then you got cancerous growth, so this one is not inherited mutation, right? So this one is a mutation on that specific area that you can cut off, and then you are cured of the disease, the cancerous growth, for example. But then if the mutation occur in the sperm and ovum, and then the person that has the mutation in the sperm or ovum got married, produce a children, then that uh, that child has a stable mutation that you carried, that is obtained from the parents. Okay, so that is why they cross it. So basically what they did was that they create lots and lots of mutants, and then these mutants, they grow on the complete medium, because when mutants grow in the complete medium, they can thrive, they can grow normally, isn't it? Because you remember, if some of these are mutants, they can grow normally in the complete medium, because the complete medium produce everything that can be taken in by the neurospora in order for them to grow. But then if this same mutant, many mutants, okay, they grow in, they grow in minimal medium. So I take, for example, one neurospora from here, I inoculate in this tube. If this neurospora can grow, meaning to in the minimal medium, meaning to say this neurospora is not a mutant. Okay, but out of this, many neurospora, when I transfer each of them into the tube, some of them cannot grow. So when they cannot grow, it means that this tube contain a mutant. Okay, so out of many X-ray, probably when they grow in minimal medium, you don't know yet which one of these contain mutants. Okay, but when you transfer each of them into the minimal medium tube, some of them will grow normally, meaning to say that these are not mutant, but some of them will die inside here. The one that cannot grow inside here, so when you refer back to the original tube, so you know that in this tube, it contains the mutant. So now you have a collection of one mutant. Okay, and then they grow, they do another round of mutation. Okay, probably next round they found that, okay, this tube cannot grow. In this sample cannot grow in these mutants, meaning to say that this tube contain another mutant, for example. So they have many collections of mutants. And then how do you want to know what is the mutation for this one for? So then the next round of the experiment is that you take the sample from this, which you know a mutant, and then you grow in different amino acids. So you take one of these, okay, you grow in a minimal medium, definitely it will not grow because it is a control, right? Minimal medium do not have something else, then you know that it will die. So this one is a control, meaning to say that you are still having mutants over here. But this one, you grow in different kind of tube that is containing additional. For example, we have how many different amino acids? The one that you know of? How many different amino acids we have? Huh? How many?
20.
20, correct. So you can have 20 different, oh, tengok sini pun ada. Okay, never mind. So you have 20 different amino acids. So you have, you prepare minimal medium, and then each minimal medium, each tube, you add in different amino acid. And then you take this one from here, you inoculate inside each of these, and then you will see which one is growing. Meaning to say that if it is growing in this tube, meaning to say that this mutant unable to produce tryptophan. So that is why it require additional tryptophan inside here to grow normally. So then you know that this one is a mutant for tryptophan. And then they do another, they have another mutant. So each tube, if each mutant tube, they have to need to prepare 20. So if you have 20 or 50 mutant collection, each mutant have to be tested in minimum 20, actually 22 lah, let's say, but let's say 20 tubes, meaning to say you have how many, so you need thousands of tube in order to determine each individual mutant that you have, a mutant for different amino acid. Okay, so that is what what Biddle and Tatum use in the experiment. And then they can also add in another, in the minimal medium, they can eat another compounds and so on. So based on that, they assume that many genes interact with Neurospora crassa, and based on that, each mutant, when they add certain compound, minimal medium and O-acetyl homoserine, then you can see that it can grow. Once you have this positive, the other one can grow. So based on this, they can have a series of steps to determine based on the how it can produce the growth in the minimal medium plus all these things. So you can, based on the result, you can have a series of chemicals that was converted for the enzyme to produce a normal individual, for example. Okay, so based on that, they determine position of the gene. So that is how it comes to one gene encode for one enzyme for Biddle and Tatum. Okay, and eventually it was revised to one gene, one polypeptide, because an enzyme can consist of more than one polypeptide. Remember biochemistry, STB1063 kalau tak silap. Remember that? You can have, for example, this one is an enzyme. Okay, nampak tak? Okay, I'm going to stop for a short while because I can see that my face is not, my movement is not detected by the camera. Okay, bear with me for a short while. Can someone tell me whether my screen is moving or not?
No, doctor.
No, yeah. I don't know why it stopped. Okay, I'm going to. Okay. To share again. Remember one. You are at the one gene, one polypeptide, isn't it? Yes.
Yeah.
No? Okay, one gene, one polypeptide. Why is it revised into one gene, one polypeptide? Because you remember in your biochemistry, the enzyme or an enzyme can be more than one polypeptide. Ingat tak? Can you remember that? Yes, no, all right. Yes. Hello, anyone? Okay, let me see. Okay, one gene can be more than one polypeptide, right? So you have and those polypeptide, okay, this one is an enzyme, okay, can be from more than one polypeptide. Polypeptide 1, polypeptide number 2. This one come from different gene. This one come from different gene. Only when they are together, it forms a functional enzyme. Okay, so if one gene encode for one enzyme, meaning to say that these two polypeptide comes from one gene, which is incorrect lah, because this one come from different gene. This one come from different gene. Only when they are together, they form a functional enzyme. Okay, so that is why one gene, one polypeptide. So you can have polypeptide form together to form an enzyme. Okay, so one gene, one enzyme is incorrect. One gene, one polypeptide, one gene, one polypeptide. So that is the correct term. All right. So you can have a series of enzyme that comes from different genes in order for the enzyme to be to produce from a substrate, produce a product, and that product become a substrate for another enzyme, and this enzyme produce another product, product C, which then become this product C become a substrate for enzyme C to produce product D. So this one is a series of enzymatic reaction in which if the enzyme, if one of the gene is not functioning or mutated, then this enzyme cannot be used to convert the uh, the substrate C, which is the product of enzyme B. This substrate C cannot be converted into product D. Then it will have accumulation of C in the individual. Okay, whereas in the normal individual, this C is not accumulated because it is converted and so on. Okay, so that is the how the enzyme functions in the system. All right. So I think that will be the end of our lecture today. We will continue with the enzyme, the lecture, the follow up of this in the next lecture. Okay. So in the meantime, do you have any question that you want to ask about our lecture today? Do you have any question that you want to ask? At the moment, no one answered anything. So yes, anyone? Any can I? Oh yeah, thank you. Okay, thanks for the reminder. Hold on, yeah. Share. Share the attendance. Right. So